Bismillahirrahmanirrahim, and welcome back to this next video. And this is the 8th video of the chapter 2 of Immunology. We are talking about the granulocytes, and in the granulocytes we are talking about the neutrophils. So I've told you that the neutrophils, they belong to the granulocytes class of the white blood cells because they have granules in them. Now, I've told you that these granules, they come in a variety of the flavors, and we are focusing on the primary granules from the last couple of videos. And I've told you that the primary granules contain a lot of antimicrobial components. So first we discussed about the lysozymes, we discussed about the myeloperoxidases, we discussed about the defensins, which are the important antimicrobial components present in the primary granules, sometimes also known as the Azurophilic granules of the, you can say, neutrophils. Now, in this particular video, I want to focus on another important component, another important antimicrobial component that is present in the primary granules of the neutrophils, and that is the elastase. Now, when you talk about this elastase, that actually belongs to the serine protease family, and by that I mean that when you look at the structure of these enzymes, the serine is one of the most important amino acid in the active site of these enzymes. So it belongs to the serine protease family, and it belongs to the subfamily of the elastase. What this elastase do is that it usually targets the uncharged amino acids like the glycine, the alanine, and the valine. And you will see that why these amino acids, these uncharged amino acids, are important when we talk about the immunological functions of the elastase enzyme. Now, this elastase, what it do is that it is going to break down the peptidoglycan layer, sometimes also known as the myelin. So the elastase is going to act on these peptidoglycan layer. It is going to break it down through its proteolytic activity. So as you can see, this elastase, that is a proteolytic enzyme. That means it is going for the breakdown of the proteins. Now, if you look at the structure of the peptidoglycan layer, so the peptidoglycan layer is actually a chain of the alternating N-acetyl glucosamine and N-acetyl muramic acids. And we talk about the importance of the lysozymes as far as the NAG and these NAM and their connection with each other is concerned. So in this particular video, as we are talking about the elastase, which is actually a proteolytic enzyme. So these peptidoglycan layer, this NAG and the NAM chains, they're actually cross-linked by short peptides. And these short peptides, they provide the overall stability and strength to the peptidoglycan layer. As you can see over here, this is the NAM, this is the NAG. As you can see over here, they are connected to each other through beta-1,4 glycosidic linkage. As we see in the last video, that lysozyme that act on those particular beta-1,4 glycosidic linkage. So the NAG and the NAM, then the NAG and the NAM, they are going to make these chains. These chains are then connected to each other by the beta-1,4 glycosidic bonds, and the lysozyme is responsible for the breakdown of those particular linkages between the NAG and the NAM. So that is what lysozyme is doing. Now, if you look at this one, the NAM, one NAM in one chain that is connected to another NAM. You can see over here, this is one chain of the carbohydrates. This is another chain of the carbohydrates, the NAG and the NAM. Again, you've got the NAM and the NAG. So one name from this particular chain that is connected to another name by this particular short peptides. If you look at the composition of these peptides, they actually contain like this. L is the L-alanine. This D-glu actually is a derivative of the glutamic acid, sometimes also known as the D-isoglutamate. It is also written like this, like capital D with a small I. This I refers to the iso and then the glutamic acid. The third component is actually the L-lysine, and the last two amino acids, they are the D-alanine. So this chain of the amino acids, they are actually connecting one chain of the NAG and the NAM to the other one, thereby providing stability and strength to the peptidoglycan layer. Now what the this is a very important image. This image is very much clear. This is one chain of the NAG and the NAM. This is one chain that was in another chain, and as you can see over here, these short peptides, they're actually intertwining these chains with each other, thereby providing strength and stability to the cell wall. Now what this you can say elastase do, it is going for the breakdown of these chains, and you can see over here I told you that elastase, it acts on the alanine, and you can see a lot of alanine over here. So what it do is that it is going to act on this one. This is going to break the peptide bonds, and if this chain of the amino acid, if that is broken down, you can simply see if you broke down these particular chains, this particular carbohydrate chain is going to move away from this particular chain, this one is going to move away from this particular chain, and the strength that the peptidoglycan layer derives from the interaction of these amino acids, that would be lost. When that is lost, that means that the overall stability of the peptidoglycan layer, that is gone. And when that is gone, that actually leads to the formation of the osmotically unstable cells. And when the bacterial cell wall is compromised, in this particular case, it is going to get compromised because of the function of the elastase. And in my last video, I've told you that when you break the beta-1,4 glycosidic bonds of the NAG and the NAM, that is also going to lead to the structural instability of the cell wall. So the mechanisms, they are different. So the osmotically unstable cell, the osmotic pressure inside the bacterial cell become unbalanced, and the cell may eventually burst due to the influx of the water. So when a lot of water that enters into the bacterial cell, that is going to burst down. And when that is burst, that means that would lead to the death of the bacterial cell because of the weakening and the rupture of the bacterial cell wall, because the cell wall have lose its structural stability because of the breaking down of these amino acids because of the elastase. That means that would lead to the death of the bacteria. So if you like the video, please subscribe to my channel, hit the notification bell, do comment, do ask me question in the comment section if you are not clear about anything, and share it with your friends. And we will continue the discussion, and in the next video we'll be talking about the cathepsins, which is another important antimicrobial component present in the primary granules of the neutrophils.